All right, so we will, so our, this course is about applying principles of physics to the life sciences. Okay? So the first chapter is an introductory chapter. Okay? So to understand how physics sheds light on biological processes. Okay? So we want to understand biology from a physics perspective. And you'll see what this means. Okay. So here's an MRI image. Okay. Do you guys know what MRI stands for?
Magnetic resonance imaging?
Yeah, magnetic resonance imaging. Okay. And what you do is you okay. So what is this? What is this guy? I'm sorry? It's a hydrogen atom. Okay. And do you have hydrogen atoms in your body?
Yes.
How many?
Yeah.
Yeah. So you're so H2O, we are essentially a bag of water. Okay. Every water molecule has two hydrogen atoms. Okay. And this is a proton, this is an electron. Okay. And every proton is spinning. And magnetic resonance imaging uses those protons. Okay. So those hydrogen atoms are everywhere. And we'll see how this image is taken. Okay. So here is a dialysis machine. Okay. Anybody work with a dialysis machine? Okay. So this, do you guys know what the dialysis machine does? What does it do? Yep.
Takes the blood out and then you can also put it back in.
I'm sorry?
You can take the blood out and also put it back in. Just form of it.
No.
Take over the kidney function temporarily.
Yeah, take over the kidney function. Okay. So, so let's talk about this a second. To understand how this dialysis machine works, what you want to know is, and we'll talk about this process in a little bit, you want to know what diffusion is. Anybody know what diffusion is? I'm sorry? Right. So you can take, let's say, you might take a glass of water, put a drop of ink here in the water, and what will happen? Eventually, all the ink molecules will spread out. Okay. So what is happening is molecules from a higher concentrated region are spreading out to a lower concentration region. Okay? And that's the process of diffusion. Yep. So in your body, you use that process wherever you have to transport stuff over the distance of a cell. Okay, so for instance, okay, so here are your lungs. Okay, so here are the alveoli in your lungs. You breathe in air. When you breathe in air, this air has higher concentration of oxygen than your blood. Okay, the blood that comes in from the veins to the lungs. And the oxygen from the alveoli diffuses into the blood. What else happens?
CO2 diffuses out.
And so CO2 has to diffuse out. Why would CO2 diffuse out?
So you don't explode.
It'll diffuse out because the concentration of CO2 in your blood is higher than in the air there. Okay? So CO2 is a stuff you want to get rid of. So CO2 is waste, you're getting rid of that. molecules go from higher concentration to lower concentration. And this oxygen goes from here, from a higher concentration to a lower concentration in the blood. Okay? So diffusion is the process how molecules are transported. Okay? The thing is, diffusion is a slow process. Okay? So by this process, you can only transport molecules a small distance. Okay? So if you had a big cat, for instance, or a mouse, you can't diffuse oxygen from here to there. That would take forever. Okay? So diffusion is a process where molecules move from a higher concentration to a lower concentration. All right, so now let's come back to the dialysis machine. And again, I'm going to give you a sense for this again. What I want you to do is just sit back and think about everything you thought. Say, okay? The exams are pretty stressful. You just have to carefully read the slides and stuff like that. What I want you to do is understand this stuff. All right, so what is the purpose of the dialysis machine? It's temporarily replacing your kidneys that are not functioning properly. Okay. So here's stuff. Here's your blood vessel. And here, okay, so this is a blood vessel, this is a membrane, and this is the dialysis chamber, if you want. And I'm just keeping it simple again. So what we want to do is get rid of the bad molecules. Okay? Like urea and stuff like that, and your kidneys are not functioning and you want to get rid of that. So what is it that you do? So your blood has a higher concentration of urea. This dialysis liquid has lower concentration of urea, and urea will diffuse across the membrane. Okay. Is there any problem with this? You achieved your goal. Can you foresee any problem with this? Yep.
Doesn't it need to pass through the membranes with the osmosis?
I'm sorry?
Doesn't it need to pass through the membranes with the osmosis?
That's when water molecules pass through, that's osmosis.
It's a replace the vital minerals and everything else as well.
Right. So, okay, you lost the bad stuff, but nothing is preventing the good stuff from going across. This is glucose molecules. And if the glucose molecules have lower concentration here, they'll go away too. When your kidney is functioning properly, it spends energy to pull those back. Okay. How do you prevent the good molecules from going away in the dialysis machine?
You put more in.
I'm sorry?
You put more in.
Very good. It's very simple. So all you do is make sure that the glucose concentration here is the same as in the blood, and you won't lose any glucose. Do you guys understand that? All right. So that's going to be our perspective. We want to understand how things work. Okay. All right. So that's the dialysis machine. Okay. So physics laws are fundamental. Everything in the universe has to obey physics law. Okay? So this fellow here, his body is not breaking one physics law. Okay? So his body has to work within the confines of physics laws. Okay? So all living organisms are subject to the laws of physics. Okay? So meaning okay, so here's an example. Energy conservation is a law of physics. What does that mean? What is that? What does the law of energy conservation mean?
Energy is not created or
Destroy. Okay. So the total energy is constant. Okay. And so no matter what process goes on in your body, it has to obey that law. Okay. Energy conservation. Okay. Then there's laws constraining fluid flows. Okay. So here's a simple law. So let's say here's a pipe like this. All right. And so let's say 2 gallons per minute are flowing in, some kind of liquid is flowing in, 2 gallons per minute is flowing in here. Okay. And the flow rate is the velocity of the fluid times the area of cross section. Okay, so the flow rate, and again, you don't have to remember any formulas. I'm putting up formulas so you understand stuff. Okay? So flow rate is A1, the area of cross section of this pipe, times the speed of the fluid. All right, so here's this pipe whose cross section is changing. There's no leaks in the pipe. 2 gallons per minute are flowing in. How many gallons are flowing out? What's the? I'm sorry.
2 gallons a minute.
2 gallons a minute. There's no leaks, so the flow rate there is 2 gallons a minute. So the flow rate in is A1 V1, and the flow rate out of the pipe is A2 V2. All right, you'll see in a second why we're talking about this. Again, what is A2? Is A2 bigger than A1?
Yes.
Yes. So what is V2 compared?
Velocity. Velocity?
Yeah, compared to V1 is smaller. Okay. Well, why do we talk about this? So here, let's go back to our slides. All right. So these are red blood cells, and so they have hemoglobin, and they get rid of the carbon dioxide and they absorb the oxygen. Okay. This is a diffusion process, and if the blood flows too fast, that diffusion process wouldn't have enough time to take place. Okay. Now, when your heart pumps out the blood, the blood is coming out of your heart is coming at about 1 foot a second. Okay, this is how fast the blood is flowing. If the blood flowed that fast here, there wouldn't be enough time for diffusion to take place. So what's your problem now? What do you have to solve?
Reduce the heart rate.
You have to slow down the blood.
You have to reduce the fluid.
Yeah, you have to. No, you can't reduce the flow rate.
To reduce the velocity of the blood by
Yeah.
increasing the area.
Yeah, you have to reduce the velocity of the blood. The blood is flowing here. It's flowing at about 1 millimeter a second. The blood is flowing that slow. Okay, and now you just learned how to slow fluid flow. Did you guys understand that? So how do you do this? Your blood vessels, they are broken up into capillaries, billions of capillaries. And that's how so now the area is the total area of all those capillaries. The total area of all those capillaries is thousands of times bigger than that, and the blood slows 1,000 times slower. Okay. Did you guys understand that? All right. All right. ask questions if you don't understand stuff. Yep, go ahead.
What did you say was kind of what to slow it down?
I'm sorry?
You said capillaries or something like that?
All right, so I'll do two diagrams. So here's a pipe whose area of cross section is changing. The flow rate is so A1 V1. Flow rate, how many gallons a minute of fluid are flowing, is A1, the area of cross section, times the velocity of the fluid. And if this pipe has no leaks, what is flowing in is flowing out. Okay? So what's flowing in is flowing out. So if the area of cross section increases, the velocity will decrease. Okay? So what you take is you take a big blood vessel in your body and break it up into lots of little capillaries, and all the area of all the capillaries add up to a large area. And that's how you slow down blood. Okay? All right. Okay. So all living organisms have to obey the laws of physics. So we just showed you how laws of governing fluid flow determines what your body has evolved into. Okay. So here's an image, okay, and physics provides tools enabling image and measure cells and okay. So these are, I guess, viruses. Okay. This is a scanning electron microscope image showing. So these are salmonella invading immune cells. Okay. So here is a scanning electron microscope. Okay, so and you can see that it can take images with resolutions less than the size of much smaller than the size of a virus. Okay. Yeah. Sometimes I'll tell you about how the scanning electron microscope works. Okay. Do you guys want to hear about that now? Has anybody worked with a scanning electron microscope? Have you seen one? Have you heard of one? Okay. All right, so here's how this thing works. All right, so let's say you want to take the image of something, okay, and all objects have bumps and stuff like that. Will have bumps. Okay, so on the scanning tunneling microscope, you take an electrode like this with a pointed stuff. Okay, so here's the object whose image you want to take. And what you'll do is, so you'll take this electrode and scan the surface of the object whose image you're taking. So that's where the scanning part comes in the name. Okay. So I'm going to draw the electrode. Okay. And then what you do is, between these guys and this, the object you're taking an image of, you connect 25,000 volts or something like that. I'm just making up a number, a large voltage. Okay. So what happens is, when this thing is far from the surface, so when you apply this high voltage, if the gap is small, you'll have electrons tunneling. Like in Harry Potter, you go through the walls, you tunnel. Okay? So the electrons will jump from here to there. When the gap is small, okay, so the probe maintains its distance. Okay. When you hit a bump, When you hit a bump, more electrons will jump across. Okay? So you're measuring the current. As you're scanning it, all you're doing is measuring current. A higher current just means a bump. And this image, all this stuff is just proxy for the current that was measured. Whenever you see a bump, that was higher current, and when you see a depression, that was a lower current. Okay? So scanning is you're scanning the electrode. Tunneling, what did that word tunneling refer to? The electrons tunneling through the gap. And microscope, it's an instrument that sees tiny things. Yep.
I understand the concept of graphing,
Scanning.
scanning the process and taking that data of varying resistance.
Varying currents.
With the three-dimensional imaging and the colorization, how do we get to the three-dimensionality? I mean, it's not normal,
Well, it's not a real image. The colorization is fabrication. And then
Yeah, so he's got a good point. So what you get is a black and white stuff. This is a different structure, and so you know that was a different structure, so you just gave it a different color. 3 dimensions. I don't know how that works. I think it's a flat thing. It's a. But that's how the process works. Okay. So our goal in this course is to help understand living systems and biomedical tools. Okay. So we want to understand how everything works. Okay. I want you guys to ask questions. I might not have all the answers, but I'm going to send you down rabbit holes. Okay, that's my goal. Biological processes are physical processes. They have to obey physics laws. Okay? Physics can help you understand a great deal about biology, biochemistry, and medicine. Okay, so let me tell you, so here's an example. What is this thing? What is this picture?
Which one?
This one, the top one.
Zappa?
blood vessel.
So it's a capillary, and you can see that here's this blood vessel as a single red blood cell going through the capillary. And this is for a reason. Okay. Remember, why did you have capillaries?
For diffusion.
I'm sorry?
For diffusion.
For diffusion. Well, you also wanted to increase the area so the blood slowed down and stuff like that. So the blood is flowing at about 1 millimeter a second or something like that. I'll give you the exact number. And so diffusion can happen. But this width of the capillary is not an accident. All right, so let's say if the capillary was slightly wider. I'm sorry? I can't hear you. Okay, yeah. Oh, about slowing down the blood? Okay. Remember we talked about, so this is a pipe with varying width, and if the pipe gets wider, the flow rate is the area of cross section times the speed. So if 2 gallons a minute flow in here, 2 gallons a minute have to flow out. Okay. So if this blood vessel got wider, the blood would get slower. Right? So that's what the body does. You have an artery, and this artery breaks into billions of capillaries, and all this area adds up to a lot more than this area. And so the blood is flowing much slower in the capillary. Okay? Blood is coming out of your heart at about a foot a second, and when it comes to the capillaries, it's flowing up about a millimeter a second. All right. So now we'll talk about the width of the capillary. Yep.
So, like, this, like the left side, is
Mm.
the velocity is higher because it's smaller,
Yep.
and then as it opens up, it gets lower.
Yep. You've experienced this. Have you used a garden hose to water? Okay, so water is flowing in the garden hose, and then you want to spray further out. What do? You make it smaller. You make it smaller, the speed increases, and that water goes further.
So, okay, so if it's going faster, it goes further, right?
I'm sorry?
So if it's going faster, it goes further.
Yeah, right. Yeah. All right, here's another thing. This is the only business where we take the money up front. So ask questions. You've already paid for it. Okay. We're happy if you don't ask questions. We got the money already. No, I'm not happy. I'm just saying. All right, so now we'll talk about the width of the capillary. Okay. So you saw that the capillary was this wide. Okay, one red blood cells can fit through. If the capillary was a little wider and maybe one and a half red blood cells like this would go through. Okay. Now, to make this blood flow through, so you have to push on the blood. It's not frictionless, so you have to push. And here, the pressure is higher, the pressure is lower here. And the pressure difference is what pushes the blood through like that. If the capillaries were this wide and let's say 1.5 cells could fit through, this pressure difference would have to be a lot higher than here. When the red blood cells are going single file, it's easier to push through. The pressure difference is not that high. What does that mean?
More efficiency.
Your heart is not working as hard. How long does your heart have to work? All right. So you can have efficiencies. If you improve efficiency, it benefits the environment.
Won't that also increase, like, the molecular diffusion efficiency by touching both sides?
Right. So it was solving two problems. Okay. Biology has the best solutions. Okay. How come biology has the best solutions? It has millions of years to evolve to find the best solution. All right, so you guys see when these blood vessels go, red blood cells go single file, you don't have to push as hard, you don't have to work as hard. Okay? And also, this allows the blood to slow down a little more and diffusion to take place. Okay? The circulatory system from the heart to the capillaries is governed by fluid dynamics. Okay, so we'll talk about fluid dynamics in chapter 9. Neurons signal each other via electrical pulses that travel along axons. So we'll come to that in a second. Okay. So here are neurons, and they talk to each other via electrical pulses. Okay. All right, so here's an image that you might have seen, and so we'll learn how this image is taken. Okay, so we'll talk about green fluorescent proteins. Okay. Have you guys heard about these things? That's what's used to take these images. Okay, so for instance, okay, so let's say there's this protein, and you want to see where this protein is in your body, in the cells. Okay? And you want to take an image of that. Okay, so in this image, so there, see, wherever that green light is, that's where the protein is. Okay, its concentration. So what you do is, you take this protein and attach a fluorescent molecule, and I'll tell you in a second what this fluorescent molecule is. Okay, attach this fluorescent molecule, and that protein goes to wherever it is going, and now what we'll do is we'll take an image. We'll see where that fluorescent molecule is. Okay. And now we'll know the location of that protein. Okay, so let me show you how we'll take an image of that. Okay, so here is this fluorescent molecule. Okay. Okay, now before I do that, I just want to all right, so let's okay, what is this? It's an atom. It's a helium atom. It has 2 protons, 2 neutrons. Here are the 2 electrons. Helium is the top right corner. It's an inert element. Okay. So these are the allowed energy levels. Okay. So it's got an infinite number of these levels. So there's the helium atom in the ground state. Okay. Now, what you can do is you can excite this helium atom. If you shine a light, this is a particle of light called a photon. So if you shine a light of the right color, you can excite this electron to there. This atom can absorb the helium atom, the photon, and go from here to there. So now that's an excited atom. Okay. Now, generally, this atom is excited, it stays excited for about 1/1000th of a second. What does it do? I'm sorry? This is not fluorescence yet. So this will fall back and send the exact same photon. Yep. And this generally happens in 1/1000th of a second. Okay, so you learned how here's a molecule. You send a photon, it got excited, and then it emitted the light. Yep. So we are going to use molecules like this. Okay. You guys following this?
Is that photon is the iron or like a gadolinium? Those are the photon?
No, photon is a photon is a light particle. Okay. The energy of the photon is a constant. This is a Planck's constant times the frequency of the proton. higher the frequency, more energy the photon has. Blue light has more energy than red light. Okay? So the frequency of blue light is higher than the energy of the red light. UV light has more energy than infrared light. X-rays have more energy than visible light. Okay, you guys understanding this? Yeah. All right, so remember what we did? What are we trying to do? I don't want to lose you guys. We are trying to understand this image. Okay? All right, so that image was an image of, let's say, some kind of protein and the location of those proteins. So what did we do? So you can't just shine light like this on a cell. Everything will reflect that light. You just take an ordinary camera, everything will reflect that light. You can't, so you don't know where that protein is. If you want to take the image of something specific, what you're going to do is you're going to attach these tag molecules. Those are the fluorescent molecules. Okay, and now let's see what we'll do. All right, so in this example, so here's when the atom is in the ground state, here's the energy level. When this electron went there, this was one of the energy levels of the atom. And the atom has many energy levels. I'm sorry?
Go ahead.
It's not screen coming up on the screen.
I know, it's been doing that. Okay, I don't know what's going on, but at the beginning of the semester, there's always some glitches. So you'll just have to let me know. Okay, so every atom has many energy levels, and here are the energy levels. You saw how to go from here to there. How to go from here to there? You just have to shine a photon of the right energy. Okay? And when it goes back from there to there, it just emits that photon. Okay? Okay. Now, those fluorescent molecules, so the fluorescent molecules, they have many more energy levels. Okay, so here is the ground state, and you see the ground state has so many sublevels. Here is an excited state. Here's many excited states. So what you do is you shine a high energy photon on that molecule. It absorbs and goes there. I'm sorry? You shine a high energy photon, and the molecule, that tag molecule, gets excited from here to there. And then the property of this molecule is it immediately loses some energy, part of this energy as heat. It loses that within a trillionth of a second. And then, in a billionth of a second, it goes from this level to that level. And you see, this is less energy, so this light emitted is a different color. All right, so what did we do? So here was this molecule. You shone UV light on it, and only your tag molecules sent out green light. Did you guys understand that? You took a picture of exactly the molecules you tagged.
So the excitation process changes the wavelength of photons?
No, well, when a photon is absorbed, a complete photon is absorbed. When the energy levels are different, you can lose part of that energy in a different process. So you lost, so this photon was absorbed and you went up till there. From this excited level, you lost some of the energy as heat. And then you went from this level to that level, and this energy was different, and the photon wavelength was different. So you will see images like this. So you can tag one molecule with green fluorescent protein. You can tag another molecule with red fluorescent protein. All kinds of stuff. Okay, so that's how those images are taken.
So the fluorescent light
Mhm.
that comes from UV lights. when
So
you
the
shine-
you
right,
you shine UV
you
light.
shine- Yes,
Mhm.
you shine the UV light on it, you see the fluorescent whatever on the object. That's the light that's stronger.
No, it's actually weaker, less
Okay.
energy.
It's different then.
Yeah, it's different. It has to be different because it's coming from the tag molecule. If it was the same light is coming from everywhere, and you wash out the whole image.
Alright.
Okay.
So we measure it based on the emission, not the fluorescent
Yeah,
light
yeah,
is
yeah.
the same.
When I take
but
a
it's
picture,
emit-
I'm looking at a reflected light. Yep.
Yeah,
Mm.
the emission is different to each molecules. Emission done by each atom is different, that's how we measure.
No, the key was this was the light that you shown on the sample, and you're looking for this particular light, this which was different.
Emission.
Yeah. Yeah. Yep.
So is it only one molecule
So in this image, this protein, there's the protein, that's the molecule that was tagged, that was what was absorbing the UV light, and that was the molecule that was fluorescent. Okay. And then see the tag molecule. And there's billions and billions of those tag molecules inside that organism. Okay. All right, so that's how that image is taken. Okay. These molecules, they occur naturally as well. Okay, so there are molecules that fluoresce in your body. Yeah. Okay, do you guys have any questions on this? I mean, this is, if you understand that, that's neat. And that was worth it just for showing up for today. Okay, in my opinion. Yeah.
So, just like, so I'm understanding this correctly,
Mhm.
relaxation is when it's higher energy and.
No, relaxation is when it's emitting the energy, when it's falling down to the ground state.
Yeah, when it's like
Mhm.
falling down and it doesn't emit, fluorescence or light, the emission is light, no, it's.
Okay, so this is absorption. Relaxation is, okay. Excitation is this process. Relaxation, the excited electron quickly loses some energy via vibrational relaxation. Okay, this is relaxation. It's losing some energy via heat, actually vibration. Okay, that's heat. This molecule bumped into its neighbor and gave some of this energy, not via photon, but just bumping in. And then it went from here to there. That was emission. Okay. So excitation, relaxation, and emission. Yep. Yep.
Is the whole molecule emitting the light or just that?
The whole molecule. Mhm. So every molecule has various levels. Okay. So it absorbs within a trillionth of a second, it loses some energy, and then in a billionth of a second, it emits the photon. Okay. So the key is the emitted light is a different color than the absorbed light. And so you know exactly where that light is coming from. Where is the light coming from? From the tag molecules. Okay. All right. So you have, again, like I said, all the answers are in the slides. You just have to read them carefully and make sure you understand stuff. Okay, Stokes shift. The emitted fluorescence is a longer wavelength. The emitted fluorescence is less energy. Okay, so here's the speed of light, frequency times wavelength. If the frequency decreases, the wavelength increases. Okay, so for any light, here's the wavelength. Okay, as the wavelength, bigger wavelength has lower energy. Okay, the energy of a photon is hf. Okay. H is Planck's constant. Planck's constant. Again, you don't have to remember any of these formulas. You just have to understand stuff. Okay. So red light has longer wavelength than blue light. Red light has longer wavelength than blue light. It has less energy than blue light. Okay. So you see, this is higher energy, lower wavelength, lower energy, longer wavelength. Okay. So the emitted fluorescence is a longer wavelength and less energy than the absorbed light, UV light. Okay. Fluorescence happens very quickly, usually in a nanosecond, and this is the bottleneck, and that is happening within a billionth of a second. So fluorescence is common in biology, in organic molecules, okay? Especially molecules that have rings. Fluorescent dyes, green fluorescent proteins, and other kinds of stuff. So this stuff won Nobel Prize. Yeah, this technique won a Nobel Prize when it was invented. So detection of bodily fluid in using UV light. So if there's microscopic quantities of your bodily fluid someplace in a forensic place, there are fluorescent molecules. You shine UV light, and you can detect those fluorescent molecules. So in identifying minerals, ink in currency and document. Okay. So there's fluorescent ink molecules in currency. Okay. So you shine when you've seen people put currency notes under a light, they're putting it under UV light. Yeah. Okay, we'll talk about one more thing. Oh man, this is going slow. Okay, so we'll see how this guy works. Everybody's used this, right? What's that?
The pulse oximeter?
Yeah. Have you used it?
Yes.
Okay. As a job or as a patient?
Personally.
Huh?
I own one.
You own one.
For personal use.
All right. Okay. So, physics explains how sap rises through the xylem in increase that can be 100 meters tall. 100 meters is roughly a football field and a half length. I mean, so it's not easy to explain how water rises in a redwood tree from the ground all the way to the leaves. Okay. I don't know, have you guys heard of the capillary effect? All right, so let's say you take a glass of water, you take a thick straw and put it there, put it in the water, and there's no water rising, okay? What you'll find is, if you put a thinner and a thinner tube, thinner tubes are capillaries, the thinner the tube, more the water will rise. Okay? The water sticking to the walls is being pulled up. Again, this is the capillary effect. The thinner the tube, the higher it'll rise. These water channels in trees called xylem, they're thin, but they're not thin enough to raise the water 100 meters. Okay? So the question is, how do the redwood trees get the water up all the way to the leaves? Okay, so that's a problem. But we'll come to that later. Okay. So now we want to understand this instrument. It's a pulse oximeter. Like the name suggests, it measures your pulse rate. It does two things. It measures your pulse rate, and it measures the oxygen level in your blood. Okay? And we'll see how it does. So you stick a thin body part, a finger, or they can attach it here as well. Okay. And here's the deal. So here's the blood vessel. So that's the pulse oximeter. What you do is you send two beams of light, okay, from the bottom. One is an infrared beam, and one is a red beam. Okay. Now, so here's the notation. This is hemoglobin, and this is oxygenated hemoglobin. So the red blood cells with hemoglobin which has oxygen attached to it. Okay, so and here's hemoglobin, which doesn't have oxygen attached to it. Okay, what do you want? You want every hemoglobin molecule carrying oxygen. You want that to be efficient. So you want 100%. Okay. So here's the deal. So at the bottom of this device, there's those two. At the bottom, it's emitting those two beams, and at the top, you have detectors. All right. So at the bottom, you have the two beams that I talked about: infrared beam and a red beam. And at the top, you have detectors. Okay, so infrared light, so the wavelength of infrared light is 940 nanometers. You don't have to know that. Red light has this thing, wavelength of 616 nanometers. Infrared light is absorbed more by oxygenated blood. Okay, so if more of your blood has hemoglobin has oxygen absorbed, this beam gets absorbed and the intensity of this beam will be less. If the blood has more unoxygenated blood, hemoglobin, this gets absorbed more and the intensity of this will be less. So all you have to do is measure the intensity of these two beams and you know what fraction is oxygenated.
Is there supposed to be something on the screen?
Yeah.
Okay.
Did you guys understand that? So you use two different, I'm sorry?
This wasn't on here, so
I'm sorry?
this wasn't showing up.
Okay. You want full value for your money, huh? All right. So here's infrared beam. The infrared beam gets absorbed more by oxygenated blood. Okay, the hemoglobin that has got an oxygen molecule. So if more of your hemoglobin has got oxygen attached to it, this beam will be less intense at the top. This beam, the red beam, gets absorbed by unoxygenated hemoglobin. So if your blood didn't have much oxygen, this beam would be less. So you measure the intensity of these two guys and you know whether your blood is oxygenated or not. Okay? So that's what this is. It's got two beams and it's got detectors for the two beams. Okay? And here's the definition. Okay? So this is oxygenated blood and this is oxygenated blood plus unoxygenated hemoglobin. Okay? And that gives you the fraction of your blood that is oxygenated. Okay? Of course, you want to be close to 100%. Okay? And so for instance, I wear a CPAP machine and stuff like that. You know, when you go to a doctor's office, they don't want it below 90%. So and so on. Yep.
So you said if the blood is unoxygenated, the beam will be stronger or weaker?
Okay. So if it's unoxygenated,
It's blank.
No, there's two separate
There
beams.
you go.
so you're monitoring two beams. Okay, watch this. So you have an infrared beam and a red light beam. Red light gets absorbed by unoxygenated hemoglobin. Okay? So if there's a lot of this, the red beam will get weaker. And if there's a lot of this beam is not affected at all. Okay? So for any detector to work, you have to have two different things that behave different in different situations. That's the idea.
The
Okay.
accuracy.
I'm sorry?
It's for like you said, two beams to use the accuracy. If
No,
it's-
two beams because each beam is behaving differently whether the blood is oxygenated or not. Okay. Yep. R is the red beam. IR is the infrared beam. Okay, what does the pulse oximeter do?
Measures your blood saturation and your pulse.
Yeah. So now we understood how the blood saturation worked. Now we have to understand the other function. Okay? How do you think it works?
Same way that you can take your own pulse.
Right. So when your heart pumps, the blood goes in pulses, so to speak. Okay? When the heart pumps, when the blood is flowing, more blood is flowing, this blood vessel got bigger because more blood was flowing. It's not a continuous flow, it's a pulse flow. When more blood is flowing, both the beams get absorbed more. So the intensity here is changing as a function of time. It's going up and down as the blood pulses through. And there's your pulse rate. Very smart. Did you guys understand that? Yeah. All right, so again, so normal SP saturation levels, so natural saturation levels are 95 to 100%, okay? All right, so we will stop here today.